My PhD is in the reproduction field. It's dealing with the preterm labor. At that time, we'll be dealing with the progesterone controlling the smooth muscle contraction at the end of the term. And then I married this concept of progesterone interact with RNA splicing, bring into the androgen field. That's how I end up here in Prostate Center. So I was recently invited for review on oncogene to understand how the RNA splicing regulate the cancer cell phenotypes and in terms of pathology and physiology. And after over review the whole literature in the field, I really find out in the normal cells, the RNA splicing play a very important roles on all aspects of gene expression, starting from the genomic DNA stability to chromatin reorganization to a pre-mRNA transcription, mRNA splicing, as well as the protein post-translational modification in downstream the signal cascade be triggered by this splice variants. So it's really well known in the protein level. We have a lot of BCA members has either pro-apoptotic to anti-apoptotic characteristics. That's well published in the literature. Relatively unknown about the chromatin remodeling that are regulated by RNA splicing. And there's new knowledges in the genomic stability is really exciting. So it could be the next phase of the study, so we can try out in the prostate field. But the RNA splicing is so robust to control each step of the gene expression. The cancer cells can also take advantage of those cell processes. And the consequences to the cancer cells result in two consequences. One is the geotoxic effect that causes cell death. There are therapies targeting splicing, and right now in the clinical trials one, I will talk about that later. And the other, more oftenly, when the tumors take on advantage of RNA splicing and alter their survival signal pathways or change their cell lineage and progress the tumors into a more advanced stage. I emphasize that the RNA splicing is such an important aspect, and I feel during the review of this process, I feel the basic knowledge of RNA splicing has accumulated so much, and there's certainly a gap for basic scientists and researchers in translational research could have involved in this particular aspect. So particularly the genomic stability is exciting because when the gene is transcribed by RNA polymerase II in the ball presented as a ball, the RNA can hybridize into the one strand of DNA, and leaving the other strand is free, hanging in the air, and that's more easily be attacked by the nuclease. And the RNA splicing process help it to prevent this R loop to be degraded or attacked. So I bring up this model just to give us a better picture is that the transcription and RNA splicing could happen at the same time. It's called the co-transcription system. So what is RNA splicing? So RNA splicing is sitting in between the transcription and the translation. For all the DNA to be transcribed into RNA is pre-RNA. It cannot be translated into protein until the intron can be removed by the RNA splicing machinery. And take the overview of the AR gene was well known in our genome. In this audience, the AR exons are tiny. Is emerged in the large size of the introns. So removing those introns and put all the small exons together is a very difficult and challenging job. Yet the cells processes every daily basis. So how does it work? So in two adjacent exons, we have a particular 5' splice size as a donor size and a 3' splice size. And usually you need help at the branch size. Usually is adenosine. The donor side or the receptor sides are very conservative, starting with the GT and the AG. It could be changed in the different nucleotide, but more overly, 99% of the chances are those are recognized by the splicing machinery. So the biochemistry reaction happens like this way. So the donor side, the 5' splice size guanine, the first 4 residues first attacking to the branch site. That starts the first trans-esterification reactions. Then in this, at the end of the first step, you form RNA lariat and the free end of the 5' splicing site. Then this free end will be attacking to the 3' strike site to complete the ligation of 2 adjacent axons together. And the lariat, DNA lariat, which is the intron, large size intron sequences, has been released and decayed. And how do we know this? Is early work we have done. Like people know there are many genes has intron axons linked together with the axon introns. Axons are mixed together under the control of the T7X66 promoters. Basically, those plasmid you can construct into a plasmid and do an in vitro transcription assays. And during that in vitro transcription assays, you can even label the transcripts, which is use P32. This is the in vitro transcription products. When this product has mixed with the nuclear extracts, contains a lot of the splicing factors, U1, U2, etc., they will generate different sizes of the RNA products. When you run on a urine denatured gel, you can see the different size migrates throughout the gel. And because then you know the sequences and the size of the gene products, you can tell by those DNA sequencing gels, you can tell which gene products we're dealing with. So this is the initial study. And then if you adding a different size of the small nuclear RNA morpholinos to block the splicing site, you can change the RNA splicing efficiencies as shown here. So the biochemistry reactions of nucleotide is performed, executed by proteins called spliceosome. It's a large protein that contains more than 100 protein components, but the core spliceosome are small nuclear siRNPs. They are majorly the U1 protein recognize the 5' splice sites, and the U2 recognize the branch sites. That's the initial stage. And then once these 2 proteins are engaged, they will recruit the further triplex protein complex called U4, 5, 6, and that initiate the enzymatic activities. So after through 2 steps of trans-esterification reactions, the RNA lariat and the protein are dissociated, and the 2 adjacent axons are ligated, and those proteins are recycled and go back to the cycles to do another round of RNA splicing job. Of course, if how does the U1 and U2 recognize specifically and precisely the splicing site? It's being helped with the RNA. They call snRNAs. So those snRNAs has complementary sequences recognize specific splice sites, and they are bound. So, for example, U2 protein are bound to the U2 RNA and the small RNA, and then use the RNA as a guide to screening the whole pre-RNA and find out the particular splice site for splicing. And this is process, like I said, is being performed by core spliceosome, but also being modulated by co-activators and co-regulators, such as SR binding protein, SR serine and arginine-rich proteins, as well as heterogeneous nuclear ribosome nucleoproteins, HNRPs. They are members in this system, and those protein cofactors, they recognize particularly exon splicing enhancers or silencers, or intron splicing enhancer and enhancers. So the compromise between these cofactors are determining the outcome of this particular exon being included or skipped into the mRNA and for translation. So there are 7 different types of alternative RNA splicing. Alternative RNA splicing is a different combination of the exons you join together for protein translation. The most common occurrence, the alternative splicing is intron retention. Is intron sequences have been inserted into mRNA. That results in different splice variants. So the consequences of those different combinations gave majorly two readouts. One readout is change the reading frame, and the amino acid sequences changed, or you get an additional insertion of amino acids being inserted from the intron sequences. The other is that because of the non-relevant intron sequences being inserted or into the mature RNA, and then they can't stand from the proofreading of the machinery, and that goes to the RNA decay and destabilized and triaged through the system. So adding to that, we have to realize almost all the genes in our human bodies has splice variants. We have about 22,000 genes encoded proteins, and there are many other genes. About 40,000 genes are non-coding proteins, non-coding genes. But on average, each gene has 3.4 isoforms of different splice variants, and that covers a spectrum. So we know our human genome are very similar to the mouse. The genome size are very similar, yet because of one of the major contributors of our system are more complicated than mice is that we have more RNA splicing occurs in our bodies and give a more complicated system than mouse. So that's why one of the explanations, a lot of clinical trials are successful in the mice and does not necessarily work out in the clinical trials. So there are 3 mechanisms to regulate RNA splicing. I will touch each of them. The first of all is the splicing sites are being selected by cis element, which is the RNA sequences. For example, exon splicing enhancers or silencers, as well as transfactors are the RNA splicing factors, as well as co-regulators or co-repressors. And the RNA splicing should be separate the reactions occurs in our body is coupled with the gene transcription. So gene transcription rate, initiation, as well as elongation does play a role on the splicing outcome. And the very lastly is the epigenetic regulation of our splicing also play an important role. So I don't mean to make this figure more complicated, but just give you an idea for a particular exons to be included. So for example, alternative splice exon to be included, it involves multiple proteins involved. They have to recognize a particular enhancers for splicing. So the enrichment of the ESE will engage those particular exons to be recognized and define the 3' splicing site and 5' splicing site so that it can be included into the mRNA. But in a scenario that inhibitors comes in, such as the second example, the FOX1 and 2 comes in, then once they're recruited to the particular exons, then they can expel all the active splicing factors out of the range of the target exons, and that will abolish the inclusion of particular exons to be included. The other scenario is the inhibitor proteins are sitting recruited next to the active form of exons and also play inhibitor roles. So all these scenarios tells us the splicing outcome is a compromise result of co-activators or co-repressors, and involves in both the protein and nucleotide interactions. This is very similar to transcription processes. The transcription factors recognize a particular response element in the promoter region and start the cofactors involved in an DNA polymerase goes in. The transcription also is coupled with RNA splicing. So back to the first scenario is that we have a lot where human genes have very large intron sequences. So the average size of the 7.5 kb. If our human genes transcription rate is ranging from 6 to 70 nucleotide per second, the average will be the 40 nucleotide per second. So that costs approximately 3 minutes, more than 3 minutes, to pass one exon, and only 8 seconds to pass through the exons. So that's very challenging for the splicing complex to recognize particular exons and to do the splicing job. So therefore, if you have a fast elongation driven by the RNA polymerase II, the weak splicing site usually got ignored because it's so fast, and then usually that happens that you have an exon skipping. But if you have a slow elongation, they basically fall into two categories as the first served, first excised. So once the POL2 pass by this particular exons, and they will cut them and jump these first exon, second exon together to include it, or it don't cut simultaneously. It cut when all the transcription finished. Then, but at the same time, the RNA splicing factors are engaged in the particular exon's boundary, so that they will be recognized and included later on after the termination of the transcription elongation. So transcription elongation rate or transcription initiation rate does play a role in the outcome of RNA splicing. So this is the work done in 2002 by O'Malley in Baylor Medical School. This is a very phenomenal work being done, and it's very simple. So basically, he put in the MMTV is the progesterone or androgen or the glucocorticoid response element in the promoter region of a minigene, as a CD44 he used in this particular assays. And if you're adding the progesterone, you turn on the gene transcription initiation as well as the elongation, and you find that the skipping versus inclusion ratio changed. So you more likely see escaped gene products versus the inclusion gene products. That's characterized by here. But if you have a constitutive active, the HSP promoter in the 5' and adding the hormone doesn't change anything. This is very hard evidence to show the transcription elongation and the initiation play a very important role for the RNA splicing outcome. And this has a particular implication of ARV7 RNA splicing, which has very clinical relevant to CRPC. The third mechanism we're talking about is epigenetic modification. So when RNA polymerase II walking along to transcribe the genes, they are not go through the same speed. There are a lot of road bumps in the middle. They're called histones. Histones are wrapped up with the genomic DNA. You have to relax it so that the RNA polymerase II can pass through. So a different modification of histones C-terminal tails determine the local speed of the POL2 to pass by particular exons. And the recent work can demonstrate that particularly histone marks can recruit adapter proteins, in this case it's MRG15, and that can further recruit RNA splicing factors. So once the gene passes along this particular exons, the RNA splicing factor already recruited by the adapter proteins and results in the, because in this case the PTB is also called HNRPI, play an inhibitory role. So that's more often result in the skipping of the exons. But in the different scenarios, you have different histone modifications. The MRG is not recruited in the system, you more likely receive exon inclusion, this perspective. So now we have at least four of those adapter proteins. They are being demonstrated associated with the splicing factors and play a role in the splicing outcome. So in general speaking, RNA transcription and RNA splicing are in the same machinery, and they perform simultaneously. And a lot of the transcription machinery protein components are form a tight complex with RNA splicing factors. So there are three perspectives of RNA splicing could play a role in cancers. First of all, the mutations, because of the large size of the intron sequences, a lot of mutation occurs in the intron sequence, and that happens inside or nearby the splicing sites that play a role in the splicing outcome. The other is a mutation of the splicing factors, has a very clinical applications, and targeting those splicing factors could lead to the therapy, anti-cancer therapy. And the other is more subtle and more complicated, is the cancer therapy-induced alternative splicing. The first aspect is from intrinsic factors. The genomic mutations cause the different splicing, but the third part is environment, the treatment of the cancer causing the alteration of RNA splicing. So this is a global genome-wide studies showing the comparing the normal tissues versus the recent cancer tissues, and they sequence the whole genome, not only the exon sequences, and they find out that the cancer cell, almost all the cancers, has mutations, and those mutations barely involved in the intron regions, and more oftenly, they involve in the tumor suppressor genes. So, for example, 53, P10, MET oncogenes are being destroyed or change the function by those splicings. This phenomenon is, at first thought, is very normal, with novel as exciting, but on the other side, it should be very rational. Think about the large size of the introns. If mutation occurs randomly on the whole genome, the more likely to fall into the intron sequences because of the large size. And the tumor suppressor being singled out because once the tumor generated, those tumor suppressor has to be released. You can't have a functional tumor suppressors ongoing and functional, and yet the tumor genesis occurs. So this is a lesson we learn, and that's what we see in our cancer genome. The other phenomenon is that the splicing factors very often to be attacked and get somatic mutations in this scenario. There are lists of the splicing factors has been documented to destroy the functions. So in here, the finding is that usually it happens as a heterozygous. That means at least one copy of the gene has been mutated, loss of function. The other relies on the other gene to be function for normal daily base uses. The impact of those could be not only related on the splicing factor itself or splicing reactions or splicing outcome. It could be trigger downstream signalings. Say if this gene has generated a splice variant, and that variant could trigger a different signal cascade down the road to totally reprogram the phenotype of the cancer cells. So I will put this example. This is well known in our prostate cancer field, is the AR-V7 splicing. So AR gene has 8 exons, an intron retention, which is crypto exons C3 are happens to split into the coding region so that they form a C-terminal truncated form, and this splice variants lost the ligand binding domain. They cannot receive the stimulation from the ligand. Thereby, people think this is one of the major contribution for the CRPC progression. So we think we know androgen receptor for decades. It's been cloned 40, 50 years ago, and the biology of AR has been extensively studied. It's until this work been identified, reported in 2012. So people realize that there are splice variants of AR gene and they play an important role in this. So what I try to emphasize here, and RNA splicing is an open area of studies and largely is under investigation, and there are a lot of things I know. So once we know there are splice variants, so we kind of working on it, find out how the gene AR gene been spliced. So this is the initial paper published about 2 years after the ARV7 being identified and cloned. And in this particular paper, we conclude a few things. First is the generation of ARV7 is through RNA splicing. It's rare cases from a gene rearrangement. There are argument that gene rearrangement is the major mechanism, but it turned out to in the tissues as well as the cell models as xenografts is not common. It's very rare system. Transcription coupled RNA splicing is the general generic mechanism. Secondly, ARV7 synthesis are determined by AR transcription elongation and initiation rate. And then we also identify the splicing factors responsible for those, and the recent work has also been demonstrated more splicing factors involved in this protein complexes and the complex works. And we also, the recent work done about the histone modification also play a role here. So this is a study in the paper. We're talking about the VCAP cells is how bearing the AR gene amplification. You have AR full length and AR variant. Basically, we treat the cells with different scenarios of the hormones. The first scenario is the DHT or DHT followed by MDV enzalutamide, or enzalutamide or enzalutamide plus DHT, follow up with the DHT. Basically, those androgens manipulate AR gene transcription rate. So if you do AR inhibitors, then you basically boost up the AR gene transcription. At the same time, ARV7 go up. We also did a treatment, so we used atmacin D and the DRB to inhibit transcription initiation as well as transcription elongation of the AR gene. And you find out the same trend of change, AR full length and ARV7 change coordinately, suggesting the AR splicing is coupled with our AR gene transcription. Using many gene, we demonstrate those particular splicing enhancers are important to determine the ARV7 splicing, and using RNA pull down, we show the proteins recognize the particular sequences for generation of ARV7. And recent work, we've been working with Martin and Jian Fei from Maryland to show that this CRIP exons, exon 3B, have been methylated by at the histone 3 and lysine 9, and this methylation is regulated in GMJD1A. And once the recruit engage of this histone modification protein, will further recruit the HNRNPF and result in ARV7 splicing. So go back to the original basics knowledge is transcription elongation rates, splicing factor recognize particular splicing sites, splicing enhancers or inhibitors, as well as epigenome regulate AR gene transcription. So therapy-induced progression also reflects into the CRPC scenarios. We see late stage prostate tumors become neuronal type of CRPC with the loss of function of ER. And it happens become more prevalent when the new generations anti-androgens are being treated to the patients. This is highly metastatic and poor survival, resulting poor survival. And it's now been known that those neuron tumors are derived from the adenocarcinoma, and possibly through two mechanism. One is through a stem-like phenotype, intermediate stem-like phenotype, but the other is through a direct transdifferentiation. So we initiated this study to generate the hypothesis that the adenocarcinoma and the APC has relatively the same genomic landscape of mutations. Their DNA are same. The structure, chromatin structure are unknown. RNA splicing is unknown, and the transcriptome are very different, totally opposite, and thereby the phenotype is totally opposite. We hypothesize the RNA splicing play an important role through two ways. One way is directly regulate transcriptome and generate different RNA splicing splice variants. The other, it can reprogram the histone structure, a chromatin structure. By doing that, we profile comparing the adenocarcinoma versus the neuroendocrine tumors, and not only analyze the total transcript, but also the RNA splicing signatures. And by two different patient cohorts and different platform and different sequencing depth of the RNA-seq, and we found a common splicing signatures contains 24 genes, and we now know that 21 of them are regulated by a gene called SRN4. It's a neuron-specific splicing factors. So the identification of SRN4 is not because of the expression, it's because of the function. So we find the function, then we find out it's the gene regulate this transformation. So what is SRN4? SRN4 is a neuron-specific splicing factors. It's important for neurogenesis and during the embryonic development. And if you have a functional impaired SRN4, you more likely have a neural disease such as autism. And the mechanism of the SRN4 is to regulate the particular UGC motif at the intron regions next to the 3' UTR, 3' splicing sites, so resulting in inclusion of tiny exons into the mRNA, and that result a totally different neuron version of the proteins to be functional. So we will be able to use the RNA hybridization technique to show that SRN4 is highly expressed in small cell carcinomas and lowly expressed in adenocarcinomas, and this expression is highly correlated with three neuron biomarkers, chromogranin, CYP, and CD44. And interestingly, there are some tumors in the middle of the transition we call the adenocarcinoma with bounded neuron differentiation. We also find certain tumors, adenocarcinoma tumor has some low expression levels, suggesting the adenocarcinoma progression to any tumor are in a dynamic way, depending on the capture of the tissue collection time point. We have different molecular phenotypes. And we also profile this gene expression in the adenocarcinoma in hormone-naive tumors as well as the castration-treated tumors. So we want to find out the key information here is that there are a small number of the adenocarcinoma cells bearing low levels of SRN4 is hibernating inside, although those cells are adeno phenotype. It could be triggered by the castration, and those tumors, because of the survival advantages, become populated. Using the RNA techniques, we not only could be able to profile particularly splicing, we can SRN4 but we can also analyze the function. So the RNA splicing splice variance is very difficult to analyze in tissue levels because the tiny changes in the coding regions. So basically, if you can use the RNA probe and recognize particular junctions between the in this case BC82, you can generate a very correlated associations of the splicing factor expression levels as well as the function of the splicing factors in the matched samples. So this is the model we propose, and each of the genes we published in the paper in a different in a separate project. Basically, we can say the SR4 is a very powerful transformer of adenocarcinoma into neuron tumors. The BIF1 is a protein regulated apoptosis. So through regulated splicing, they gave the tumor cells, adenocarcinoma cells, a survival advantages. And through a different AR or androgen receptors, REST or SOX2 and FOXA, those transcription factors, they can change the lineage from adenocarcinoma luminal epithelium cells into a neuron lineage. But that's not enough for the switch for the tumor to be developed. You need something to accelerate the tumor growth, tumor cell growth. So in this case, we know the MIP6, BC80, LSD1, they're all keystone modifiers. They don't do any transdifferentiation job. They only accelerate the cell proliferation for the tumor genesis. And to that, I will go back to the hypothesis that SR4 is important to either regulate the transcriptomes, mRNA levels, or reorganize the chromatin organizations. And this is the synchronized the proliferation and differentiation result the transition of adenocarcinoma into neuron tumors. To summarize, so clinical, I appreciate this audience has a lot of interest in the ARVs, clinical relevance of RNA splicing. So well known is the ARV7 has been used as a diagnostic tool, and the emerging knowledge is about circular RNA. I think is very important and it's critical. It's going to be a high impact in this field. So the timing of ARV7 has identified is in 2012, with the same time as enzalutamide or abiraterone being developed, probably at the same time. And this is the work done by Jun Luo's group in Johns Hopkins to show that the patients are more likely to fail the enzalutamide or abiraterone treatment if they are ARV7 positive, and that their survival is poor. So the method we have done is that they collect the blood from the patients and they capture the CTC circulation tumor cells using antibody to capture that, and then perform a real-time PCR to detect V7. And this method has been really licensed to Xuesen and then developed a commercial kit. Right now it is being used in the CLIA lab. Basically, they use magnet several beads to use antibody to precipitate the CTCs, and they have in situ PCR reactions generate. And there are multiple variations of the method. You use a real-time PCR or digital PCR, and recently they developed a ARV7 specific antibody that can do immunofluorescence labeling of the CTCs to identify ARV7. Then because of the ARV7 has no ligand bind domain, presumably driving the CRPC, the direct therapy strategy is that if you have V7 more likely to fail the hormone treatment, then they more likely give a better outcome if they go directly to the chemotherapy. That's the rationale behind it. I want to draw attention to this is I think the timing of ARV7 is identified overlap with the enzalutamide development, so that gave a lot of hype about this particular markers. But keeping in mind ARV7 is not tumor specific or CRPC specific or enzalutamide resistance specific. A lot of benign tumors or benign prostates also has ARV7. Generally, they have it, and the specificity is a problem. The other difficulty is that the CTC assays relies on the capture of the CTC. If you can't capture, you have a low tumor burden. You have less cells in the circulation system. You're not likely to capture them. At the end of the day, ARV7 is a biomarker. It's a biomarker represent association of the disease, particular disease progression stage. It should not confuse whether ARV7 is a driver of the CRPC. There are a lot of arguments right now about whether ARV7 is truly driving the CRPC. Given the concerns that the ARV7 always co-express AR full length, it's unclear when ARV7 is generated, the full length function, the mechanism of the full length function, whether there's a change or not is still remains to be determined. The circular RNA is particularly important in the field. I think there are two impact papers published in Cell earlier this year. And originally we thought the circular RNA is a byproduct, it's a waste, non-coding, there's no function at all. Because of the next generation sequencing technique, we know the circular RNA contain about 10% of the whole mRNA spontaneously expressed. Because of the circular form, is relatively resistant to exonuclease digestions. They are a longer half-lives. Usually a biomarker such as PSA is passively released into the bloodstream because the structure of the base membrane is invaded and disrupted. That's why how the PSA being released in the circulation system. The circular RNA can be packed into the exosomes and possibly repelled into the blood system. So likely, if you have a tumor-specific circular RNA, you more likely detect it earlier than the markers that passively release into the bloodstream. And the other benefit comparing to protein markers is those nuclear type markers carrying the genetic informations. For example, if you have AR gene has mutations that can be contribute to the enzalutamide resistance that happen to package into a circular RNA, and that sequences can be detected by digital PCR as well as our second generation sequencing. So it is an important field. We recently published this collaboration with Yandong's group in Tulane and find out there are multiple circular RNAs that's been synthesized by the AR genes. And we pay particular attention to the circular RNA generated by the exon 3. We call the circAR3. We can show the circular RNA can be detected in the tissue levels. It's downregulated as tumor progresses. However, in the bloodstream, if the circulation levels is increased, so that gave us a difficult time. The reviewer keep asking how come the tumor levels reduce and the circulation level increase. Our argument is that it's the release rate, it's not the expression levels determines the concentrations of blood. So, for example, PSA is highly expressed in the tissue level in the benign prostate. You detect very low levels in the bloodstream. The same thing happens here. And we know this circular RNA is from prostate or prostate organs because if you have a patient whose blood will be collected before surgery and post-surgery, you can see the trend of change decreased after the prostate or prostate organ being removed. So that tells us there are possibilities and there are high opportunities for us to find the tumor-specific circular RNAs that can be developed into biomarkers. So those blood has been collected by Alan. I think is helping with that. Martin helped with that. Peter Black also get a lot of help with that. Without the clinicians' participations, these findings cannot be found. In terms of the therapy, there are three categories. There are 5 categories of therapies you can targeting. First, you can target the splicing factor itself. For example, the H3B8800 has been developed under phase 1 trial. It's targeting the SF3B1 splicing factors. The rationale behind it is the multiple leukemia patients has a heterozygous mutations. So they only relies on one copy of the gene to be functional as a splicing. By inhibiting the splicing factor induce the genotoxic effects so that the tumor cells are more vulnerable to be tapped. The other is the antisense oligos has been used to treat various difficult disease called SMA disease, has been proved by MDA. There's always a doubt about antisense, and actually the first drug being approved by FDA is about 2 years ago. There are efforts have been concentrated on the kinase that are responsible for the splicing factor phosphorylations. Oligo-based are ongoing for many years. The first one success in 2017, and if particular splice variants are the driver gene for the disease, you can also target that directly. So oligo-based technique are, I think Martin has a lot of experience in that. In terms of splicing, you have different strategies. Either you can design the oligos sequencing complement to the splicing enhancers or silencers, or you can design the sequencing containing sequences that can saturate the splicing factors. So block either way, either the response element versus the RNA splicing factor to rewrite the RNA splicing processes. Of course, there are also challenges about efficacies of delivery of nucleotide, particularly in this case, the splicing reaction happens inside the nucleus, so you have to pass through 2 membranes to be effective. That's also been challenging. This is the disease being proved that they call the spinal muscular atrophy. It's majorly caused by this gene as SM1 genes, and the mutations or deletions just disrupt this gene's function. Fortunately, we humans have a homolog of the gene. The problem is the exon 7 has mutations that result most of the gene products is non-functional form, 10% are functional form. Through antisense rewiring, they have 2 strategies. One strategy is a design oligo contains matching sequences to the exon 7 to cover up these mutations, at the same time recruiting more splicing enhancers that will enhance exon 7 to be included. The other strategy is blocking the splicing silencers by designing oligo. Both efforts has been moved forward, and at the end, this one succeed at the end for FDA approval. So 3 learning objective this day. We go through the basic biochemistry of the RNA splicing, and we talk a little bit about the potential clinical application as diagnostic tool as well as the therapeutic for cancer treatment. Thank you.